Hello everyone. So today we are going to talk about HMP pathway. So first of all, what does HMP stands for? HMP stands for hexose monophosphate pathway, right? Then it is also known as pentose, PENTOS, pentose phosphate pathway, or phosphogluconate pathway. First of all, there is no different names by which HMP pathway is known, right? Then what is HMP pathway? So let's understand it. Okay, HMP pathway, it is an alternative pathway to glycolysis and tricarboxylic acid cycle for the oxidation of glucose, right? Then we have to know where does it take place, right? Where does it take place? Place is number one in the cells cytosol. So we have to know this point. Then number two, it takes place in liver, adipose tissues, tissues, adrenal gland, RBCs, testes, and finally mammary glands. Okay, now what is the importance? Because we have to understand what is the importance of HMP pathway, right? So we have to know that it is important for necessary for formation of NADPH. Okay, now this NADPH is required for synthesis of fatty acid, steroids, right? Now in the pathway, whenever the pathway is taking place, it forms formation of pentoses, that is pentose sugar, right? Pentoses, which is required for synthesis of DNA and RNA, right? Okay, again, since this is HMP pathway, we can say that there is no formation or use of ATP in HMP pathway. Okay, now let us come back to the example, right? Now it is also required for free radical scavenging, that is antioxidant reaction involving NADPH, right? Now again, it is necessary for synthesis of glutamate, dehydrogenase, detoxification of drugs, phagocytosis, and finally maintain concentration of glutathione required to preserve integrity of RBC, red blood cells, membrane. Okay, in other words, we can say that it is necessary to keep the F2 plus state of ion in hemoglobin in reduced state, so that with hemoglobin won't be formed. Okay, these are the various importance of the HMP pathway. Okay, now let's go towards the process of HMP pathway. How does it take place? Okay, process. Let's go one by one. It is easy to understand. So initially, from glucose, there is the formation of glucose 6-phosphate during glycolysis, right? So this glucose 6-phosphate that we have will get converted into 6-phosphogluconolactone in the presence of glucose 6-phosphate dehydrogenase enzyme. Okay, this D means dehydrogenase. Now during this process, there will be the generation of NADPH H plus in the presence of magnesium. Okay, now another thing that happens is the 6-phosphogluconolactone by the addition of water in the presence of gluconolactone hydrolase get converted into 6-phosphogluconate. Now understand that this is a 6-carbon complex, right? 6-carbon compound. This is also a 6-carbon compound. This is also a 6-carbon compound, right? Now let's look at how it changes. Now what happens is there will be the formation of 3-keto 6-phosphogluconate. Okay, this is also a 6-carbon compound. Okay, now in this step also there will be the formation of NADP H plus from NADP plus. Okay, now what happens is by the removal of a carbon dioxide here, okay, there will be the formation of ribulose 5-phosphate. Now obviously this is a 5-carbon compound. Now this reaction from 6-phosphogluconate to 3-phosphogluconate takes place in the presence of 6-phosphogluconate dehydrogenase. Okay, right? Now what happens is that this ribulose 5-phosphate in the presence of two different enzymes get converted into two different molecules. Okay, so first of all, in the presence of epimerase, it get converted into xylulose 5-phosphate. Then again, in the presence of enzyme ketoisomerase, it will get converted into ribose 5-phosphate, right? Phosphate. Now, if you consider, then or if you remember, then this thing is a pentose, isn't it? This thing is a pentose, right? This is a pentose. Now what happens is these two react in the presence of enzyme transketolase in the presence of TPP, thymine pyrophosphate, and get converted into a 7-carbon compound and a 3-carbon compound, that is ceroheptulose 7-phosphate and glyceraldehyde 3-phosphate. phosphate. Now, since this is a 7-carbon compound, this is a 3-carbon compound, right? Again, these two react in the presence of transallolase, right? And get converted into two different things. A 4-carbon compound, that is erythrose 4-phosphate and fructose 6-phosphate. Now, to remember, this is a 6-carbon compound and this is a 4-carbon compound, right? Now, then what happens is a very good thing takes place. That is, this xylulose 5-phosphate and erythrose 4-phosphate will react, okay, and then form two different compounds, okay? That is glyceraldehyde 3-phosphate, glyceraldehyde 3-phosphate, and fructose 6-phosphate. Now, again, this is a 6-carbon compound. This is also a 3-carbon compound, right? Then what will happen is this glyceraldehyde 3-phosphate, by reversal of glycolysis, will again form fructose 6-phosphate. Okay, now we have to understand that this whole process, the 6-carbon compound is being converted into 5-carbon compound, right? And this 5-carbon compound is again converted into 7 and 3-carbon compounds, and again into smaller, that is 4 and 6-carbon compounds, and finally forming a 6-carbon compound. Also, important thing to remember is that initially when this reaction is taking place, 6 molecules of glucose 6-phosphate, 6 molecules of glucose 6-phosphate is being converted into 6 molecules of phosphogluconolactone, and again 6 molecules of this is being formed 6-phosphogluconate, again 6 molecules of 3-keto phosphogluconate is being formed, and again 6 molecules, sorry, and again 6 molecules of ribulose 5-phosphate is being formed. From that, 2 plus 2, that is 4 molecules of xylulose 5-phosphate is being formed, out of which 2 molecules of ribose 5-phosphate is being formed. Now, 2 molecules of xylulose 5-phosphate and 2 molecules of ribose 5-phosphate will react to form 2 molecules of cerebrohapulose and 2 molecules of glyceraldehyde 3-phosphate. Again, 2 molecules of erythrose 4-phosphate will be formed against 2 molecules of fructose 6-phosphate. Now, the remaining 2 of the xylulose 5-phosphate will react with the 2 molecules of erythrose 4-phosphate to form 2 molecules of fructose 6-phosphate and 2 molecules of glyceraldehyde 3-phosphate. And these 2 molecules of glyceraldehyde 3-phosphate will again form one molecule of fructose 6-phosphate. Okay. Now, overall, 5 molecules of fructose 6-phosphate is formed. Then, what can happen is this fructose 6-phosphate can again be converted into glucose for glycolysis and again formation of ATP by Krebs cycle, electron transport chain, right? So why is this needed? Because this is needed in special organs, right? Like in liver, adipose tissues, adrenal gland, RBC, especially and mammary glands. Why? Because it is needed for the formation of NADPH. Now, why? Because obviously these NADPH testes and mammary gland will form their steroids like hormones, and these things can also be formed. Like in RBC, glutathione maintains the RBC patency, that is the membrane patency. And again, if we talk about the glutathione reducing system, it will prevent the formation of superoxides. So it implies superoxide of various oxidants that are being formed in our body, are very harmful for the body. So we have to understand that. Similarly, in liver, for various metabolic purposes, detoxification of drugs, as we already said here. Right. So these are now I will tell you about how does the glutathione and NADPH system prevent the antioxidant injury. Okay. So let's talk a little bit about the antioxidant action. Okay. Antioxidant action of HMP pathway. So first of all, let's understand in HMP pathway there is the generation of NADPH plus H plus. Right. Now what happens is suppose there is a superoxide. Suppose there is oxygen free radical, right, or hydrogen peroxide. Then what happens? This is very harmful to the tissues. Right. So this is converted into water by glutathione peroxidase for oxidase. Okay. Now during this process, the glutathione will get oxidized, and the oxidized form of glutathione will be formed. That is GSSG will be formed. It is the oxidized state. Okay. And the reduced form is basically GHH. Okay. And again, this oxidized state it will be converted into a reduced state by the action of glutathione reductase. Now this is mediated by NADP and DH plus H plus. So this is basically the antioxidant action. Now let's talk about some abnormalities that take place in HMP pathway. So first of all, there can be G6PD, glucose 6-phosphate dehydrogenase deficiency. Okay, now this deficiency will lead to hemolytic anemia, right? While taking various drugs like primaquine, which is used in malaria, then sulfomethoxazole, which are sulfonamides, then while also taking fava beans, this leads to favism, okay, FAVISM, favism in jaundice. Moreover, a person will get falciparum malaria resistance as champison and reduced glutathione for growth in RBCS. Thank you.